said at the Institute, I have the pleasure of serving with Freeman Dyson, who is often makes wonderfully iconoclastic statements. And one of them, which is a good introduction for this talk, is that he points out that physicists believe that they're about 100 years ahead of biologists in the quality of the data they collect, the decimal to which they can take out the data. That's all true, actually, I have to say. and so they're running about 100 years ahead. But Freeman believes just the opposite. And he believes just the opposite. That is to say, biology is about 100 years ahead of physics because of the following reason. Physics has two theories, of course, relativity and quantum theory, which at very small dimensions actually start to even contradict each other. And we're going to have to sort that out, that there are two theories which are not been unified in any way, and so this brings a certain tension into physics. But Freeman points out that biology for 160 years now has had a single unified theory. And it begins with Darwin, of course, and it's the theory of evolution. And it's the theory that will drive many things that I'll talk about today. And so let me say a few words about what Darwin introduced into biology that's become really clear at the molecular level, at the DNA level, that it appears to be really correct. Darwin pointed out, first of all, that a species, that is a breeding population that can have offspring that are fertile, a species has diversity. Right? And it generates that diversity by making a certain number of mistakes every generation. Right? And it actually generates diversity in two ways. Darwin could have never known this because actually contemporaneously Mendel was showing some things about combinatorics. But there's both combinatorial diversity, that's to say whose chromosome you pass on, mother, father, grandfather, grandmother chromosomes, right? And recombination of those chromosomes, right? So that you're passing along combinations of the parents, and so there's combinatorial diversity. But Darwin wasn't speaking about that kind of diversity. I think he was, if I was to paraphrase Darwin, really thinking about errors that are made every generation so that there are, in fact, a certain number of changes that occur as parents reproduce and the offspring inherits these errors. Right? To put it in today's parlance for diversity and errors, I'll just give you a little bit of quantitation. There are about 3 times 10 to the 9 in a human being. There are about 3 times 10 to the 9th nucleotides or base pairs that make up all the DNA, the genetic information that we have. Right? If we were just to replicate that DNA, just duplicate it with the machinery for duplication, we'd have an error rate of about 10 to the minus 4th. About 1 in 10,000 times we'd make an error. And that's right on the edge of tolerability of getting information from one generation to another. So we have really excellent editing functions which recognize those errors and correct them and get the error rate up to or down to about 10 to the minus 7th. Right? Which means that every generation there are about 300 errors on the average. Right? There's sometimes more, sometimes less. If you're making eggs or sperm under stress, there may be a higher error rate. Some people have naturally higher error rates than others, but within that distribution of error rates, let's say the mean is about 300 per generation. And that is a good way to interpret Darwinian diversity. The older the species, the more generations it goes through, the greater the diversity of the species. And then Darwin said, upon that diversity acts natural selection, to take the fittest and allow fittest for reproduction of the next generation and allow them to out-reproduce the less fit. And so we select upon diversity and we keep changing. And ultimately, we could change in a way that gives rise to a new species, which would be a non-breeding population, would start its own breeding population, and new species arise from that, usually from isolation in various ways and kinds. So that's what Darwin meant about the generation of diversity and evolution of the species. But Darwin, of course, didn't quite think in all the terms that I've told you about. He didn't know about the numbers and so forth. And it could happen, in fact, I'll take you through a little experiment now. It could happen that organisms can even set their mutation rate slightly higher. So if we take two bacteria that are related to each other, they're identical genetically, and we grow one of the bacteria in a culture condition that's very stable. The oxygen's the same, the glucose is the same, the nutrients, the environment stays very constant for 100 generations. And the other, we change a lot. We change the oxygen content, the glucose content, we change the volume they're growing in, the density at which they're growing, right? So they'll get used to changes. And then we measure the mutation rate or the error rate from generation to generation. The one that lives in an unstable environment actually has a higher mutation rate, makes errors at a higher rate. Right? And so we can set those errors. That's an important concept. Just by differences, in other words, mutations in the genes that replicate the DNA themselves, make the DNA more sloppy in copying, make information transfer sloppier. And that happens when the environment changes. And stresses are particularly good at giving you these high mutation rates. And stresses are a real problem. Right? So a long time ago, the organisms counted this diversity, this information transfer, this lack of fidelity in information transfer, by putting one more failsafe into the system, right? And the failsafe was that when it recognized that there was stress in the system, and this stress could cause mistake frequencies to go up 1,000-fold in some cases, right? It killed the cell, right? So it has a fidelity factor. It got a fidelity factor that watched over error rates that got too high and decided to enforce that by death. Right?
That's true for bacteria?
That's actually less true for single-cell organisms because death for single-cell organisms is suicide. But it's certainly true for us. For example, I'll give you a typical example of a stressful situation. People who undergo chemotherapy, males or females, right, have lots of damaged DNA, and that's true for the germ cells. And the germ cells will then undergo a programmed cell death called apoptosis, right? And you will temporarily be killing off your germ cells. The germ cells can come back. This can be repair, right? And some people will store their germ cells. In the case of humans, store their germ cells so that they are taken before the chemotherapy, right? Because of this death, this wave of death. Now that, I'll show you, that happens in flies, that happens in worms. And it doesn't have to be chemotherapy for flies and worms. It could be starvation conditions, which is a stress. And they will not lay their eggs because they're being killed, right? So these are fidelity factors. And you'll see there's a single family of genes that make up these fidelity factors. And they're kind of interesting because they're a little anti-Darwinian, right? Just as Darwin will have this distribution of errors and frequency of diversity, these are pressing in on the distribution to make it a narrower distribution of diversity because it could get out of hand, right?
Can I,
yeah, please.
The mechanism is to detect the change in the error rate, not to detect stress in any other way.
Yes, so
right?
It depends on the organism, right, as to what they consider error rate or stress. A very common mechanism from simplest organisms through humans would be DNA damage. Right? So damage in the genetic information, breaks in the DNA or inversions or whatever, but especially breaks, right? Chemical modification of the DNA, that kind of damage, when you replicate over, you make errors. Right? And when that's seen to occur, the cell is killed. Right? So that's the common thread. But as I mentioned, starvation could be a stress in worms. It's actually starvation is a very big stress in females that are anorexic, right? They actually stop producing estrogen because the eggs are undergoing programmed death. This molecule I'll tell you about, actually. When the eggs undergo programmed death, estrogen stops. When estrogen stops, menstruation stops. Right? And it's all because of this particular gene I'm going to concentrate on, or set of genes, which are killing because of starvation. It really, in evolutionary terms, no one should be laying eggs into a barren environment, right? Be the worm or the human. right?
Is the action of this cell by cell? Is the damage
Yes.
determined cell by cell?
Cell by cell, yeah. So the damage is seen internally in the nucleus of the cell, and the program is rolled out to kill of that cell. Right? So that it doesn't have to kill all cells, it only kills those that are damaged. Right? So now I'm going to go back to evolution and say, where does this start? What is the oldest, what's the most, in today's parlance, what's the most primitive organism today for which we can see this family of information transfer, fidelity genes, right? Okay, so that's really good news. It's right here. This is a simple, pretty simple family. This is a sea anemone called Nematostella, right? It's in the family of coral, hydra, and sea anemones, right? And you can see, I think, pretty clearly here. And this is composed of two cell types. It's a very simple organism. It was one of the earliest organisms to arise with just two cell types. We all are born and brought up with three cell types at the beginning of our life, and it becomes ectoderm, endoderm, right, and mesoderm, muscle and bone and so forth. Here's the ectoderm on the outside, right, and here's their gut. That's the endoderm. That's the two cell types. They have some specialized cells on there called nematocyst cells here, and they feed, and then the bacteria and the single-cell organisms pass through, and then out the other end. Right? And occasionally you'll see a cell that is a little indistinct here, right, which is actually a germ cell. Right? So it's a cell that's going to be like an egg or a sperm. It will be shed. There'll be mating in the water, right, and a new sea anemone will arise sexually. So they have a sexual cycle. They also have asexual cycles. The way you actually grow these guys in the lab is you cut them up to little pieces, and then they regenerate themselves, right? So that's the asexual cycle, and that's a lot easier than the sexual cycle, turns out. So that's the way people mostly grow them. But the only place in this organism you find this gene, this protein that protects the germline, right, that I told you, the fidelity factor, is in that germ cell. It's not in any other cell type. The single germ cell it has. Right? So sea anemones, to put it in the sort of wild evolutionary terms, sea anemones today live in estuaries. They live deep down in the mud, but they come up to feed on the single organisms and the bacteria, which are often photosynthetic and live on the top of the water. But when they come up to the top, the ultraviolet light from the sun causes DNA damage, and when that happens, the cell is killed.
But when you cut it in the lab,
Yeah.
this is not used by this organism as a stress?
The real answer is we've never asked. It may well be, and it may start, but it grows back. Not only the ectoderm, the endoderm, but the specialized cells, right? So that's a program of development that undergoes. Very simple, right? If I trace back in an evolutionary form, when this organism has arisen, and therefore would have a common ancestor with, let's say, humans, it's about 1 billion years ago. Right? So I'll just set, that's a fuzzy number, I'm going to just set for you that this mechanism, this is the oldest organism we know of, or the most primitive, right? This mechanism of watching the germ cell for fidelity is about 1 billion years old, right? Okay, so now I've finessed enough. I have to introduce you to the proteins and the genes that are fidelity factors. So I'm going to do that in this slide. Right? In humans, this is called the P53 family of genes. Right? But let me start off with, because these genes, if they're in all organisms between sea anemone and us, they are in worms, they are in flies. Right? So this is meant to be a stick figure, which is parsing out the structure of the protein. Right? So this is the picture of the protein in the stick figure, right? Linear stick figure. And it's a two-dimensional structure, right? But everywhere you see a box, the amino acids in the protein, the structure of the protein, right, is unique and has a function, right? So there are at least 3 or 4 functions because there are 3 or 4 boxes. This middle function, called the DNA binding function, right, is what the protein does. It recognizes DNA sequences, and it binds to a specific sequence, right? Now, what does it do when it binds to the sequence? It promotes the expression of that gene or its messenger RNA. It produces its messenger RNA. The messenger RNA is translated to make a protein, And this is the protein that's translated. It's the protein that's going to go to the eggs or the sperm, or in one case, it'll go to somatic cells, right? And it kills when it sees damage. It's a fidelity factor, right? Now, the DNA, we can tell these proteins are related because there's, in this brown DNA binding domain in the middle here, right? There are 3 genes in humans, therefore there are 3 proteins. They happen to be called P53, P63, P73, right? And this brown region is about 200 amino acids. It folds up and it binds to DNA. It binds to DNA next to the gene where it is going to transcribe that gene. And that gives it its specificity. And its specificity is going to be killed, right? Yeah.
Good question. You said that it kills when it sees damage, but
Yeah.
doesn't it depend upon the level of damage?
Yes, it does. There's thresholds, levels of damage. It also actually can help repair processes and things. So it's possible to actually repair the processes and then it will calm down again. It usually kills in pretty extreme cases. But I'm trying to keep it very straightforward here and not play with the subtleties too much. Right? When this says 65%, those are identities of amino acids. So out of 200 amino acids at any position, there's 1 in 20 possibilities for what amino acid would be there. 65% of them are identical amino acids in the 3 proteins at the identical position from 1 to 200. Right? That means they all came from the same precursor, right? And they're really diversifying themselves now. Right? There's about 25% identity in the first domain. That's the blue-gray domain here in the first case. And what that does is it attracts the machinery to transcribe a gene. And these are the 3 proteins of humans. Let me say P63 and P73 are found in eggs. And I'll talk to you a little bit from mouse experiments that we and others have done, what their function is in eggs. Right? P53 is found in all cells of the body. Right? And this is a kind of interesting thing that happened around fish. So as fish evolve, a new gene evolves, P53, right? And it goes not to the eggs to protect fidelity of the germline, but it goes to all cells of our body. But it does the same thing. If your skin cells, if you go out in the sunlight, not today, but on a better day, you go out and you get irradiated, you could get DNA damage. That can start a cancer, the skin cancer, right? And P53 is it's not in the germline, it's in your skin, and it kills the cell. That's why it's called a tumor suppressor. Right? It suppresses the possibility of mistakes. UV irradiation is damage, right? Chemical damage you could have, right? Hypoxia, a number of other stresses, it'll kill. And it does so in the body, right? And there are people born without P53. They're born with 1 copy, and they will develop cancer over their lifetime, usually within the first 30 years. A mouse without P53 develops cancer in 3 months. So this is a protector of mistakes.
Under
Fidelity.
any circumstances? The most
Under any circumstances. And those mice live in the best place. I can't tell you how much is the cost of those mice. This is a plea, for cost. Right? These mice, these mice live in the most sheltered of places. There's nobody hurting them at all, we think. But they will be, and it's just because, the strategy, of course, of mammals, actually everybody's vertebrates, right, is that you're constantly rejuvenating yourself, your bone marrow, your skin, your intestine, right? Today, 1 billion new cells were born in everybody in this room. Each person, 1 billion new cells. So think about how many divisions you go through and how many mistakes are made. And when they're made in the wrong place, right, or if there's an elevated rate, you've either got to kill them or you will have a problem, right? And so that's what P53 does. It's a suppressor of tumors only because it's a fidelity factor, right? Born without it, you don't have the fidelity factor, the checks and the balances. So this was the first one discovered, but then we discovered 2 others that were in eggs, that were in the germline. This is the fly, P53, right? So this is called DM, Drosophila melanogaster, P53. The DNA binding domain has 25% identity of amino acids over 200 amino acids, 25% identity of amino acids at the right position. This is the worm, P53, 15% identity. What's even more remarkable
Identity to the human.
identity to the human, that's right. What's even more remarkable is if you take each one of them and do the structure, the three-dimensional structure, the structures of the DNA binding domains are identical, and they bind to exactly the same DNA sequences. Right? So over 1 billion years of evolution, they have not changed the DNA sequences they bind to each
So
other.
you said this was the first organism which has this.
The first one was the guy behind, yeah, it was this guy.
So what happened before? So the organism which...
There must be other ways to police the system. The one way, the most homologous way in bacteria is a protein called REC-A, right, which surveils recombination to kill, it doesn't kill mistakes, it prevents mistakes. It unwinds the mistakes. So there were other ways, but this strategy of death, right, as your outcome, right, it starts here,
Okay.
right? And it continues through worms and flies and humans. And it's meant to, in all invertebrates, it's in the germline. But in humans, it also goes into the soma because there's the possibility of cancer in the vertebrates. Right? So it becomes a tumor suppressor. It was the first one found. But the germline is the old one, is the oldest one. Right? So P63 and P73 have been demonstrated to act in the germline, right, by what we can do with mouse genetics. Right? So mouse is a very good organism because we can knock a gene out. We know how to eliminate a gene from the mouse chromosome. So we eliminate the gene for P63 or P73, and we ask what happens. Right? So I'm just going to show you now some experiments, the conclusions of some experiments. Right? This is a paper back in 2006. The P63 is required for female egg production and plays a role in meiosis. Meiosis is where recombination occurs between chromosomes and lots of mistakes get made. Because you have to break the chromosome and then you have to repair it and it swaps DNA. Right? So if you didn't repair the break, you would be just like radiation damage. It would be just like DNA damage. Right? So actually, the P63 mice that are knockouts, right, are female sterols. The female makes no eggs, essentially, because there's no surveillance of those eggs. The eggs are all poorly formed because there's no surveillance. 63 was responsible for oocyte death after DNA damage. Right? So that's a chemotherapy that I've told you about. But this is done in the mouse. Right?
What's oocyte?
Oocyte is the name of an egg. That's you have a production of an egg from a precursor cell. So this is a developmental pathway in the ovary to make eggs, right? And then the eggs actually, what's thought is that in females, all the eggs are made at birth or by birth, right? And in humans, there may be 5 million eggs, somewhere between 3 and 5 million eggs. And then by the time you're 12, 13, and you become sexually mature, it's down to 5,000 eggs. And that's because of the culling of these eggs by surveillance of damage and other kinds of processes. Right?
Does that depend on P63 and P73?
Yes, it does. That's right. So P73 is interesting because it acts in a way here that's age dependent. Right? You're actually in P73, older mice run out of eggs at an earlier time. That would be called premature menopause, right? In that particular case. TA73 is responsible for what's called primordial and primary oocytes. Primary oocytes are the ones ready to be fertilized. And primordial are the ones in the precursor, right? So it's involved in both. Right? And these are experiments that have been done to actually determine what the functions of these are in mammals, right? Of these two genes. Remember I told you they're transcription factors. They bind to DNA sequences and they turn messenger RNAs on and they make proteins and those proteins function. Now there's names to those proteins. Two of them are called BRCA1 and BRCA2. Some of you know what BRCA1 and BRCA2 is. If you destroy them, you get ovarian and breast cancer, right? And they are DNA repair genes, right? So remember I said if there's DNA damage, you make mistakes at a very high rate, and this guy takes care of it, it kills. Right? But what happens if you don't repair the mistakes at all? Right? It's worse. The mistakes propagate. And so this actually transcribes the repair system. BRCA1, BRCA2, MR11, RAD51 are all homologous DNA repair systems, right? It is not only watching over the integrity of DNA, it's helping to transcribe the genes, that means make the genes express the proteins that are involved in repair.
So if they're responsible, if the P63 is responsible for pulling oocytes that are damaged,
Yeah.
in the absence of it, you have no oocytes, is that a different death mechanism?
Yes, so that's a different, there are other death mechanisms, but in the absence of it, there would be a different death mechanism, that's right, if there was one. Right. So here's an example of the consequence of this. So you see, there are women born with one copy of BRCA1. The other copy's mutated. They are predisposed to cancer. Right? Let's say they develop ovarian cancer. That's the most common cancer that they could get. So they develop ovarian cancer. In every case, P53 is also mutated. Why is P53 also mutated? It's the backup mechanism that kills when the repair doesn't work. So you need two mutations to get the cancer. It's what we would call a suppressor of the DNA repair system. If the DNA repair system is faulty and you leave damage, P53 kills. So the cancer arises from these two mutations. Right? That's another reason why we know it's a suppressor. It suppresses the DNA damage response. P73, we know it acts at a place called the spindle checkpoint. That's a place where the chromosomes separate from each other and the egg finally goes into a haploid condition, right? So it has half the chromosomes and half the chromosomes. You get two eggs, and those eggs get fertilized, right? And that's watched over by this P73. And finally, P63 mice are sterile. They're female sterile. And P73 mice are female steriles, but at older ages. Right? And that means age is a variable here. Right? In other words, mistakes, there are increased mistake frequency with age, and this surveils that, but it fails if it's not there. Right? So, and all of that is related to what the consequences are. Right? So this gave us, all of this information was starting to come through, and it gave us the hypothesis that in any population, right, there would be some people that have very good fidelity factors, right, and they would have very few errors or very few mutations in the offspring. And there are other people that have less qualified fidelity factors, and they would have high mutations in offspring. They would have the chance to have mistakes at a higher frequency. So there'd be some distribution of the frequency of mistakes, and those would all be in the variations or the distribution of differences in the P63 gene and in the P73 gene. So we don't have identical P63 or P73 genes, right? We have a variety of different nucleotide sequences, a variety of different changes, and some work better than others. Right? And when it works well, the fidelity is maintained. And when it works poorly, error frequencies are higher. Right? Now, this is a perfectly general hypothesis for what would happen in the offspring. First of all, these fidelity factors, right, would act and function in eggs and sperm. Those are very specialized cells in the body, so the parents would not have any of the disorders we're going to see in the offspring, right, which have the higher mutation rate because of the lack of fidelity. And so we would see disorders in the offspring that wouldn't be in the parents. That would be one consequence. Those are called maternal effect or paternal effect genes. They were found first in Drosophila, but they're true in everybody because they are the genes that make the eggs. They are the genes that make the sperm. They are the genes that actually go ahead and have variations in them of how efficiently it makes sperm or eggs. Yeah? And that would be perfectly valid. And secondly, the disorder we're talking about could be any disorder because it's a random set of mutations that occur as you make eggs and as you make sperm. So it could be in any number of disorders that would be downstream, right?
Right.
Yeah.
Does the expression of P63, 73, or 53 vary with age?
It does, actually. This has only been done in mice. It hasn't been done in humans. But in what it would be very old mice, that is to say, mice that are older than 2 years, they might live 2.5 to 3 years, it starts to decline. However, the efficiency with which it functions, we can tell, especially if there's a stress with a mutation, is declining at earlier ages. The efficiency with which it's working.
Thinking about the variation in various disorders with age.
Yes.
Do you have a hypothesis about what's the clue to the age of the individual cell?
Yeah, hypothesis is the right word. It's not fact, right? I mean, just the entropic aspects of the efficiency with which things are functioning, right? Over time, you accumulate mistakes. Those mistakes then are not efficient, right? So getting an egg from a 45-year-old woman is not equivalent to getting an egg from a 20-year-old woman, right? Even though they might have been made at the same time because of the entropic stresses that happened over those years. The same thing is true with sperm. I mean, for instance, the error rate, I'll talk about this. This will be my next slide, in fact. The error rate in segregating chromosomes, right, where you get trisomies. You get an extra chromosome in a particular embryo, like trisomy 21, Down syndrome, right? That error rate, right, goes up with age. Right? It's a well-known fact. The error rate goes up with age. So here I could do that experiment, right? So the most obvious place to demonstrate in humans, right, that the P63 and in the genes it transcribes are going to play a role, right, in the offspring, the embryo, is to go to in vitro fertilization clinics. Right? So we went to 2 in vitro fertilization clinics, one in New Jersey, one in New York, right, and did a variety of experiments to demonstrate that P63 had a variant, right, a genetic variant of P63 in the population, right, which was making mistakes at a higher rate. And this variant was at very low frequencies. It's about 3% frequencies in the population, right? But what we saw was this variant showed up at the in vitro fertilization clinic at about 9% or 10%, right? So if I just took the population at large that had no problems with fertility, it was at 3%. If I took the population from an in vitro fertilization clinic, it was at 9 or 10%, right? So here is the variant for TD63. And these variants are given names, very long numbers, because there are about 3 million variants between any 2 people, right? And there are about 10 million variants in the population at large, probably growing number, right? Because they haven't done the whole population at large across the world, but the ones they've done, right? There are about 10 million variations, not in that gene, but in the whole genome and in everything. And so here's a variant, and this is the way the experiment was done in this particular case in New Jersey, was the eggs that were in a freezer, right? These were eggs donated by women who had already had children by in vitro fertilization. They wanted no more children, but they had eggs left over, right? This is this problem or question of eggs that are left over and what to do with them. These women signed consent forms to allow these eggs to be looked at in an experiment because they don't need the eggs anymore, right? So the eggs are taken out from each of, in this case, 84 patients, 84 women. Each donated 4 eggs. They were fertilized, and they were genotyped. The women were genotyped at the P63 gene for this variant that I'm going to tell you about, right? So we knew the genotype of the women, right? And these women are selected to actually, because they're in the in vitro fertilization clinic, their frequency is up around 10 or 12%, 9 or 10% in that population. Right? And then what is looked at is the embryos are allowed to develop because they develop to a certain stage, and then whether or not they have triploid chromosome, whether they've made a mistake, right? The error frequency, right, is high, right? And the P63 gene became statistically significant under these conditions, and so did a gene called topoisomerase 3. Now, this is a gene that's actually transcribed by the P63 gene, and topoisomerase relieves the tension in DNA when it's unwound. right? So it's part of the process of recombination. Right? And these recombination mistakes lead to the segregation errors of ploidy, right? And so what's done here actually statistically is that some women give rise to as many as 4 different embryos with triploidy, and other women 0. And there's a gradient, and there's an excellent correlation across that gradient with this particular variation. So this is the beginning of evidence, and we have there's other experiments like attendance at in vitro for fertilization clinics and so forth. The beginning of evidence to suggest what we see in mice is true in humans. The P63 gene is playing a strong role in mistakes or abnormalities, in this case ploidy. But along with ploidy, there also are other variations and mutations that occurred, right, in these embryos. Right? Yeah?
Are these embryos chimeric? Is there chimerism and things like that?
No, the embryos actually. Well, I think the way the experiment is done is you take actually a single cell, you take the DNA, you put it on a chip, and then you see a whole chromosome have an extra copy, right? It has 2 copies of one set of alleles.
Different cells from the same embryo?
That isn't done.
Sorry, what was the question?
The question is, are they chimeras? Are half the cells triploid and half the cells diploid?
Oh, okay.
Yeah. Are they chimeras? When was the mistake made? Is it made back at the egg, or is it made at the 5-cell stage or 7-cell stage, or what? Right? So it's really a question of, the errors can occur at various times in development, because you take them through to what's called the blastocyst when they're small little balls, and then terminate the experiment, as per the wishes of the family and so forth. So this and other experiments, I'm not going to go into the details, suggest strongly that in vitro these genes show up as having variants that don't function very well in vitro fertilization clinics. But as I mentioned to you before, it should be the case that it's not only the in vitro fertilization clinic is maybe the first place they show up, right? But there may be other kinds of disorders in which these genes play a role. Right? Now, a while ago, Mike Wigler made a really interesting hypothesis. The data for autism, for example, suggests that autism is largely, probably not totally, but largely a genetic disorder. These data come from, it's been challenged slightly recently, but come from dizygotic versus monozygotic twins. And something like 7, if you're a monozygotic twin, which means that you have come from the same egg and the same sperm, but at one 2-cell stage, they separate and then they make 2 embryos. But then you can see they're genetically identical at that point. And when they're monozygotic and genetically identical, if one gets autism with a 70 to 90% frequency, the other will develop autism. But if you do the same thing with dizygotic twins, which are 2 eggs and 2 sperm, right? So they could be brother-sister, they could be as different as any 2 kids in a family are, right? The frequency is around 6%, 7%. So that was one line of evidence that it was genetic. It had strong genetic components. And Mike Wigler made the hypothesis that it was an error in this process of oogenesis, spermiogenesis, early development, right? A genetic error occurred there so that the parents didn't have to have autism to have the offspring have autism, right? Could be also that it's multiple genes that had come together, but that there was a mutation that was found in the offspring that wasn't in either parent, which is called a de novo mutation or change. And these are good candidates for de novo mutations or change, because in this case they're occurring in the egg development. There are similar genes that occur in sperm development that we're not following here. Right? And Mike went on to show that that was in fact the case, that one could see that the offspring had a de novo alteration, genetic mutation, that was not in the parents. Right? And this mutation or change he found was what's called a copy number variation. That means a piece of the DNA is missing. So you have only 1 copy of it. Or you could have 3 copies of it. Right? And that's a kind of mutation, deletion or duplication, called copy number variations, which contributes to the diversity of the offspring and the population. Right? And in fact, if you look at just everybody's genome, we're all carrying copy number variations because we all have, this occurs at some rate in the population. It's in that kind of error. And some of them look, most of them look neutral in they're drifting in the populations. Maybe they're being selected for slowly against these holes in the genome, right? But some of them land in genes which make sense that they're playing a role in the central nervous system, right? So they're candidates. They're good candidates for genes that could contribute to autism. Right? And many of them, or some subset of them, occur again and again in experiments. And that starts to be a better reason to think that there are good candidates. Right? So for us, that was an interesting thing. Mike and a group at Yale, with data from the Simons Foundation, right? There's 1,100 individual families. Right? You have mother, father, and offspring, with one offspring having autism and the other offspring not. Right? And so if it's a de novo mutation, you want to find that mutation in the offspring that has autism, not in the one that doesn't, and not in the parents. Right? And out of that group, Mike and the Yale group demonstrated there were, what's the number, Mike? 160, 200?
7% of the autistic kids have that sort of thing.
Right.
8% of the siblings.
Right.
Say that louder.
What was it? 7
versus 2.
7% versus 2%. In other words, there's an enrichment in the children that had autism. Right?
Enrichment of de novo.
Oh, de novo of copy numbers. And actually, that is the exact same number that we calculated from a different population called the agree population. That was 900 families. On average, there were 7 per 100 that had de novo changes in the agree population, while the normal population had 1 or 2.
And here it was 9% and 3%.
This was the in vitro fertilization clinic, right? So what we did was we took the data that Mike generated and we asked the question of the offspring that had copy number variations, did the mothers carry this particular allele and therefore were generating mistakes at higher rates, right? So we could look at Mike's data for copy number variation. They were validated numbers, actually. They not only came from the little chips that we use all the time, which have some noise in them, but they came from validating those chips. And those are the best validated data we had, and we found an enrichment. Now, the way we do that is just shown on the next slide. I'm just going to show you the way we do it, because this is a statistical trick that we learned is in the literature. People do this a lot. This is the work of Jörg Gott. I don't know if many of you know from he was at Rockefeller, and Josephine Ho. And they made an interesting point. They said across any gene, like P63, there are many changes. Everybody carries many differences between us, right? And they may actually contribute in the sum of these changes, but some always travel together because they're very close and recombination never separates them, and some are independent. So you want the independent ones, first of all, because independent events count more than events that are coincident, right? And you also want to look at the sum of those alleles, right? So here we have, this is the way we did it. We considered the parents of families with the de novo mutations that we got from Mike's data, right, the cases. And then the controls were the rest of the parents were controls. So the rest of the parents didn't have offspring with de novo, and we called that the control group. So we used the Simons data as case and control, right, in this particular case.
Well, they could have had de novo.
They could have, but they had not been detected.
Not been detected.
But this would be of a certain scale or size, or that's right. They could certainly. Right? And that makes these data a little noisy, but nonetheless, you'll see that it works out. We find the subset of single nucleotide polymorphisms different, with a p-value that's not really statistically significant, but it gives us the candidate genes. Here we go from 118 differences in this gene, right, and 45 in this gene, right, down to 17 and 3, right, which gives us this allele frequency in the population. From the subset, we find a set of independent, that's the unlinked, right, ones. That means they are independently segregating so that they're separate, they're independent. And there we get this down to 10 and 3, right? And then when we do a 10,000 test permutation, permute the data, and then do a Monte Carlo simulation, right? We do 10,000 independent tests, we can get the null hypothesis out and the p-value here, right? So this correlates, that's all it is, an association, the presence of these SNPs that we see in the in vitro fertilization clinic, right? It's enriched in the parents that have had these mutations, right? Now, we wanted to go a little bit farther than this. These mutations occur randomly across the genome, right? And they'd be at a higher frequency, so we wanted to ask, could they also be responsible for cancers, right? Now, that turned out to be a tougher problem, because if you think about the data set we need a data set that has parents and offspring, then the offspring have to grow up and develop the cancers, right? It turned out that just at this time, it was about 6 months ago, 8 months ago, a study called the Framingham study became public. Framingham study is a heart study. It was done in Framingham, Massachusetts, and they have 3 generations of people. And they did SNP chips for these people. They did parents, offspring, right? And they followed them over years and years, right? And they recorded all of their clinical events, right? So we wanted to ask the question, were the women who got, so we have to find a way to segregate, since 1 in 3, this is really a bigger complication than autism, 1 in 3 people, 1 in 4 people develop cancer over their lifetime, right? Not all are genetically predetermined to develop cancer, so there's a lot of noise in the system. But that's not true for people under the age of 50. So these genetic predispositions tend to show themselves as cancers under the age of 50. Right? And when that happens, it tends to be, not always, but it tends to be a genetic predisposition that could do it. Right? So we wanted to look at the age at which they develop cancer as one of the variables, and we made the following prediction. If a woman developed breast cancer at the age of 40, then she might have a genetic predisposition, and that group, under 40 developing breast cancer, ought to be in the group of parents that had this particular variation that we saw in autism and we saw in the in vitro fertilization clinic, right? It's a totally independent data set. It's another way to look at the data, and it's pushing the data into a new disorder, right? Okay, so let's take a look at what we see, right? These are the frequencies with which we see this minor allele, right? In males in blue, and in females, right? In red. This is the allele of P63. This is the variant of P63, which is predisposing to autism and predisposing to in vitro fertilization. It's by association. It's not causal. It's always a correlation, right? The hope here is, that's why I'm belaboring it, the hope is if you have enough independent data sets, you begin to feel that the association is more meaningful, right? And that's why we're doing it. This is the age at which they develop cancer. So look at this. This is the allele frequency. In this case, for this particular allele, it's in P73, right? It's at about 1.8% in males, right? And then in females, it's up at 9% if you're under the age of 45. Under the age of 50, it's here. And then it drops here. And then it continues at a higher frequency in this population, but it's highest at the beginning, right? And that suggests to us that under the age of 55 or so, these cancers have a strong genetic bias, predisposition. And that goes down because they're diluted by other cancers that aren't so genetic, but they always stay higher in this particular case. Right?
How many more or less people are there?
I'm going to show you the numbers. There's 60 or so. You'll see the enrichment factor. It's about twofold. Oh, no, so the p-values are right on borderline, right? The error bars, well, there are, I haven't got them in. Yeah, I should put them in. You'll see the statistics in a second, right? What's nice is there's a second gene called MR11. I mentioned it before. This is a DNA repair gene transcribed by P73. It's, again, related to the P73 gene. This comes down to a frequency, right? But the early ages again are high. So two
Those two
independent.
things are uncausally connected, the first one and the second.
No, the second one is made by the first one. It's a gene transcribed by the first one. You would have predicted that they both should be this way. Because if the first one is high, the second one ought to be high. But this is a SNP that's in this gene. So they are causally not linked. They're two independent SNPs. Right? So this is a gene that does the repair. If it does the repair badly, the early onset is seen. Right? And this will give you the numbers here. So the women under 50 that have the minor allele, or 14 out of 30 cases here, and it's 12 out of 60 cases over the age of 50. Right? So it's about a twofold frequency of enrichment for that group that comes together, and its p-value is about 0.05. Right? And this is, I think, because of all the noise in the system. There's a lot of noise in this system, but it's consistent with what we've been able to see. So
Do the two variants tend to be together in individuals, or do they represent
no,
two different?
they're independent. Well, there are individuals with them together, but they're mostly independent. They segregate independently, so it's about 50%.
Two different genetic
That's right. They're in two different chromosomes, actually. Yeah.
What about paternal?
Yes. So, I think I'm going to give you a hypothesis that comes from the mouse again. We think P53, the one I've left out here for germline, does the paternal effect genes. Right? And we think that because of the mouse data, right? What we see big mistakes in mice that don't have P53 in transmission of different alleles to the offspring, right? In male mice. So that's a hypothesis that comes from the mouse so far, and we're trying to check it in this case. We have to look at the paternal allele. So the way to confirm these associations, the way to try to confirm them, is to do the mouse experiment. Because in the mouse experiment, we can actually knock out the gene, right? And which is what we've done. And then look at copy number variation in offspring of the mice, right? And that would then confirm from the mouse what we suggest is happening. I call this a good hypothesis now in the females. So I would tend to stop here if that's a good time and take any more questions. I don't know how.
We can do that. We're relaxed about time,
Okay.
but if this is a good place, then it's
Yeah.
fine.
Is there any explanation for gender bias in the offspring? Will male get more mutations than females? That shouldn't be coming from P63.
There's a strong gender bias. Yes, is there a gender bias in the offspring? If I stratify the mutations into males and females, right? Is there a gender bias? And the answer, we use BRCA1 and BRCA2. BRCA2 does cause male breast cancer, but at a low rate. BRCA1 doesn't have any effect on males. The experiment we're in the process of doing would be to repeat the experiment Mike did for autism, but with the cancer patients, right? So could it uncover a tumor suppressor gene in breast cancer, right? In other words, the deletion, right? And we do have one male who had a deletion, a de novo deletion from the parent that had that allele. And that male got a cancer, but not breast cancer, of course. Had a BRCA1 mutation across an exon.
So you got a BRCA1 mutation and then a loss of heterozygosity.
It's never been reported that mutation in males should give rise to cancer, and so we don't know what to make of it, but at least that's what we saw.
In mouse, suppose you knock out P73 or something,
Yeah.
and then you look at male.
So we can't knock out.
Repeat the question.
Yes. So the question was, as I mentioned, we're gone back to mice now to confirm the hypothesis that this comes from this. And the simplest way to do that experiment is you can literally knock out the gene in mice, right? So it's not even functioning. That's very different than this. It should be a much more severe case. But when you knock it out, you don't make eggs at all, so you're a female sterile, right? So we're doing one copy, right? And we look at copy number variation, and then we look at the offspring, and we see what happens.
Do the polymorphisms or variants of P63 affect the DNA binding, or is it the levels of the protein?
Yeah. So we don't know that right now. I mean, we can easily find the P63 from such cases in lymphocytes because people save lymphocytes from people who have that particular case. And we're looking for DNA damage in lymphocytes that might have some effect, but we haven't seen much yet. But that would be another path to take, just as you've suggested, because it would be, it should be the case that the polymorphism has some functional consequence on the protein structure or function in some way. Yes?
Suppose your hypothesis with respect to the cancer turns out to be true. Could
Yeah.
you then imagine some new gene therapy that one could...
It's not gene therapy. This could be, yes, sorry. The question was, suppose that the hypothesis with respect to cancer turned out to be true, would there be a gene therapy for this? And the answer is no, right? Eggs are all made already. Sperm is not all made already. So it's not gene therapy. It would just be a warning to watch out for early onset cancers, and maybe you'd go more often to be screened, right? But this doesn't occur at a very high frequency, if you notice. I mean, so we're really talking about, this is like a probability argument, right? It doesn't account for all cases of the cancer in the Framingham study, or doesn't account for even all cases of mutations, right? So it accounts for some subset, and this plus other things, I would imagine, may play a role in egg. This is a hypothesis which plays off of Mike's original hypothesis and starts to name genes as possible candidates. That's really what it does. Yeah. Yes?
Does the flattening out of the rate of mutation in the last data set you showed suggest that there's at least one other protective mechanism?
Oh yeah, there must. I bet there are a lot of other protective mechanisms. Yes. The fact that flattens out, in one case, it flattens out at a higher rate than males, leads me to believe that there still are some genetic effects at older age, but that they're diluted by other mechanisms, right? Which may not be genetic. I mean, that is to say, not spontaneous mutations occurring during a lifetime cause most cancers. But some predispositions do exist, of which a subset is known and a subset is not.
Yeah, if we were relying on luck, then it would still be a decreasing curve.
That's right. I think that's right. Yes. We rely on luck a lot, yeah.
So in these tumors, what is the contribution from genetics and what is the contribution from mutations, somatic mutations?
We can repeat the question.
Yes, so in these tumors, can one calculate the contributions from genetics and one calculate? So it depends on the age, of course, right? So at young ages, the contribution from genetics is higher. It's, let's say, 1/3, right? And that implies that there isn't any other mechanism. That's just this mechanism we're looking at. So I think there are other mechanisms to give you a genetic transmission, and they're probably bigger than 1/3. But let's say these data say 1/3, right? However, as you get older, that's diluted, right? And ultimately, I would say, the curve for the incidence of cancers goes up exponentially, starting at the age of 55. I would say that starts at the age that I'm younger than when it goes up. but it really starts at the age of about 55 or 60. It starts to go up and is moving up exponentially. So at that point, I think somatic mutation plays a bigger and bigger role.
So, but they're like the monozygotic twins
Yes.
that you were saying.
Yes.
Is there something similar with cancer?
Yes, there is. And the frequencies, frequencies can be 25-30%, and that shows about 1/3 of the cases might be, in breast cancer, about 1/3 of the cases might be genetic.
That was my question for
Yeah.
clarification. When
Yeah.
you say 1/3,
Yeah.
1/3 of all those cancers at that age.
Correct.
So the other
2/3
are either some other
a different mechanism that's
or
genetic
some
or some non-genetic
environmental
mechanism.
or-
That's correct. That's correct. In other words, what we found that counts for is associated with, I wouldn't say it counts for, that's too strong, but is associated with about a third of the cases we see. Yes, all cases at the age of 45 or 50, right? Yeah. Yes.
So your examples of errors were all fairly gross errors, like ploidy
Yes.
or missing links.
Yes.
Is there any evidence that these
Yeah.
can act like syntax checkers for
Yeah,
stuff
it's
that
a,
looks like it's well
it's a good question. So,
And is it checking a reference copy, or
Yeah.
might you letting through evolutionarily successful
Yeah.
alternatives?
so the way to answer your question is by sequencing.
You might need to repeat the
Yes,
question.
so, sorry. So the other side of the room always gets stuck, yeah. The question was, the examples I focused on were big errors. They were deletions, copy number variation, ploidy changes, right? What about these simple errors like point mutations, single base mutations? So the actual way to address that, if we had an accuracy measurement, right, if it was accurate enough, would be to sequence both parents and sequence the offspring, right, and ask the case if, now you can tell where each of the chromosomes come from, where the recombination events are. In theory, you could paste the offspring together from the parent and subtract out the differences, right? And that would see single base changes. Unfortunately, the accuracy with which we sequence, right, is a higher error rate than the error rate we make, right? Biologically, right? So the companies that make the sequencing machines have a higher rate at reproducing than we ever have at making offspring, right? So there are sort of ways to try to sort that out. What you do is you sequence twice, right? Or you keep looking at repeated copies of the sequence and average across it, right? But each of those has a certain amount of noise in it, and so it's a daunting task. We've got actually some experts in the audience who try to get at this kind of question, but it's a pretty daunting task to do trios by sequencing and actually then say this change has clearly occurred, and I've seen it enough times to believe it, and so forth.
And do you think it's checking the original, or do you think it's letting evolution happen?
It's checking the original or?
It could be doing a checksum,
Yes.
or you're doing a syntax check.
Yes.
And maybe stress leads to increased change rates because the species needs to evolve faster when it's in a stress level.
Yes, and that may be a fortuitous fact of what stress does. That is to say,
Or by design.
but yes, but in the reason we like to avoid it in humans, so this question was maybe stress actually has a good thing because it raises the mutation rate to get around this variation in environment. That's like the bacterial experiment I talked about at the beginning, right? Maybe that's true. And that could be true, but the consequence can be pretty detrimental. If the error rate, usually if you have an error rate, like people who are born without P53, have an error rate that happens and sticks because there's no correction, right? They will inevitably develop cancer. So the consequence isn't great for the person. But sometimes they escape it for 40 years, 50 years, and it's just a matter of rolling the dice because of that. Yeah.
I thought the question was a different one, namely, how do you know what's an error?
Right.
Oh, well.
So since I asked that question an hour and a half ago.
Would not cause evolution, where syntax checking might allow viable alternative code to survive.
Yes. So how do you know what's an error, right, is all dependent on what the parents had and the accuracy with which you can see it, right?
And whether you
And
change
that-
the activity.
That's right.
There's probably a change in an error, isn't that?
Oh, well, there are neutral mutations, if that's what you're asking. There are mutations that have no consequence that we could tell. Yes, Mike?
He's asking, how does the cell know what's an error?
Yeah.
Not how we know.
Oh.
Two base pairs that don't match. Which one's wrong?
Yes,
No, but
yes.
how do you know that this is
So let me. So you want to know if you make a copy that has an error, how does the cell know which is the parent and which is the offspring, right? And the answer is it marks the parent chemically, right? So in bacteria.
Understand your answer.
No, I'm going to try to give you the understanding of my answer, right? In bacteria, the A residues, right, the nucleotide A is marked by a methyl group, and that's the parent, right? In us, the parent has a methyl group sometimes, in certain places, upon the dinucleotide CG, but the offspring doesn't, right? But the offspring becomes a parent, right, at some point, right? And that's when this enzyme comes along, sees the CG, and puts the methyl group on the GC, right? And then the offspring becomes the parent. Right? So that parents can be marked chemically by enzymes which tell the difference of parents versus de novo synthesis. I see. You asked a more sophisticated question than I interpreted. That's right. I apologize. Yes, but yes, you can tell parent from offspring. The cell knows that, right? Because of the quandary you've anticipated, right? How would you know who was the real parent, right? How do you know who your parents are? Is the question you're asking. That's right.
The wise child.
That's right. That's right. Yes.
So this is sort of a follow-up question, I realize, to their question. Is there conceivably a selective advantage to having a mutation in P63? Because it seems like it has such a high frequency that
right.
there must be.
Right. So it could be, and in theory that's yeah. Sorry. Could there be a selective advantage of having a mild mutation in P63? The distribution of diversity would go up, and undoubtedly in times of wild environmental change, it might be good to have a greater diversity in the species. Why? Because that selection of them, a cut of them, would actually survive. That helps the species survive, right? It could be that it's selected for, but that's all that's the Darwinian environmental effect, right? On that, it would become the norm in the population to move the error frequency up if there was wild changes in the environment. So global warming may have those people breed at a higher rate that have higher error frequencies, right?
No, but
to put it, yes.
But you started the lecture by pointing that there is
attention.
Combinatorial
yes, diversity. Right.
And there is diversity as a result of that.
Genetic diversity, right.
It seems that you're making an assumption that this is a result of an error. The diversity which is due to error. Is it obvious that it is not a result of
combinatorial
diversity?
It is not obvious. I mentioned, especially in the case of autism, it's easier to look at in the case of cancer, but it's a very reasonable hypothesis is that parents carry alterations, mutations, right, which by themselves have no phenotype. But when they come together in a combination, then give rise to a disorder, right? And that remains a hypothesis, though we're yet to uncover such genes, right? And rather, what Mike and his colleagues have uncovered are de novo changes that might add to that. I mean, but it still would be part of the mechanism, right? It doesn't eliminate the combinatorics that we've said could exist in the population at large, right? So 1 in 70 people, I don't know how many people in this room, but 1 in 70 people carry a mutation in the gene that causes cystic fibrosis. But it's recessive, right? And as long as it keeps moving around the population, right? And it's only when two come together that you see cystic fibrosis.
Maybe we should do one more question and then retire to the other room for more questions. You choose.
Yeah, yeah, go ahead.
So I wasn't exactly clear. Is a gene like P53 or the protein, is it directly detecting errors in the DNA, or are there upstream proteins that do that and then activate?
Right, upstream proteins. So the question was, what actually detects the error in the DNA, right? Is it the P53 protein that moves around the DNA? Right, not at all. There is a protein, it happens to be called ATM, right? Because it causes a disease when it's missing called ataxia telangiectasia, right? Okay, but this protein, this gene and this protein, ATM, is a protein kinase. It phosphorylates the P53 protein, and it cruises around the DNA and it looks for breaks or distortions of the helix. It stops anytime it sees a break or the distortion. It brings two molecules together. They activate the kinase, and then they phosphorylate P53 and turn it on. Right? So every stress has its own pathway that comes in. Right? There's lots of stresses of different kinds, Hypoxia is one stress, no lack of oxygen. There's a whole pathway that activates moves to, but it's the central clearinghouse for stress from all these different pathways that move in.
So you can see that this discovery of P53 is enormously important for a variety
It's useful.
of reasons. So why don't we go
It's made me a living.
That's a good note to end on.